Oh man, cool shirt, Graham. Where'd you get it? I have two of those. They've been retired to night shirts.
I think Erica says she's waiting to be let in.
Oh, there she is.
Hi, Erica.
Hi, how are you going?
Good to see you. Well, it is 10 a.m. US Pacific on my clock. We will wait for 1 or 2 more minutes and then we get started.
Excellent.
Welcome to all of you who are connecting. We'll be starting in just a few minutes. All right, well, it seems the numbers have stabilized, so might as well get started. Welcome, everybody. Welcome to this seminar organized by NfoLD. NfoLD is the Network for Life Detection. It is one of many research coordination networks organized by the NASA Astrobiology Program. NfoLD is particularly interested in bringing together scientists and technologists around the life detection question, how best to do that in terms of strategy, in terms of technology. And so our group here is focused on bringing those communities together and brainstorm on these fascinating topics. Today, we're very lucky to have Dr. Schwieterman and Dr. Erica Barlow, who were involved, if you all remember perhaps last year, the Biosignature Standards of Evidence workshop that Heather, who's here as well today, and Vicky Meadows were the lead organizers for. And so the purpose of this webinar today is kind of give you a little debrief about what happened there and describe some of the outcomes. And for this, our MC today is going to be Bradley Burkar. So Bradley, welcome. Good to have you here. And the show is yours.
Excellent. Thank you, Sanjoy. Yes, as he said, I'm Bradley Burkar, so I'm a NASA postdoctoral management fellow, and I've been assisting Heather with all things NfoLD and Lab. In particular, I've been helping out with organizing and coordinating a lot of stuff concerning the Standards of Evidence workshop that we're talking about today. And so I'll be helping with the introductions and helping out with the questions. We are going to hold all the questions until the end after all three of our speakers have gone. So keep that in mind. Feel free to enter them into the chat, though, if you would like. I will record them and highlight the questions at the end. Okay, so I'd like to give Heather a brief introduction for those that don't know who they are. But who doesn't know who Heather is? So Heather Graham is a research physical scientist based out of Goddard Space Flight Center. They're an organic geochemist with widely varied research experience ranging from paleoecology to phytochemistry to astrobiology. Heather has a BA in chemistry from Occidental College and a dual title PhD from Penn State University in geosciences and biogeochemistry. So they do work on deuterium enrichment patterns in extraterrestrial materials and the origin of hydrocarbons in space, and also provide support to the Mars Curiosity rover science team developing analog materials for instrument testing, and studies deuterium enrichment patterns in extraterrestrial materials to learn more about the origin of hydrocarbons in space. And their favorite organism is lichen. So take it away, Heather.
Thank you so much, Bradley. Oh man, I didn't know you were going to share so much about me. Now I'm blushing. Let's see, here we go. Sharing screen, everybody sees it. We're getting off to a good start.
That's good.
This is always the worst hurdle, right? Yeah, so thanks so much everyone for tuning in. We're going to be talking about what came out of the Biosignature Standards of Evidence workshop last summer in July. And this workshop was, of course, a joint joint Nexus and NfoLD project. And so Vicki and I worked closely putting this together, and we had an amazing dedicated SOC of 24 people, 24 SOC members. And I noticed that in the group that we have here online today, many people who are participants in the workshop and probably even were co-authors on the report that came out of it that went to headquarters. So thank you so much. If you were one of the many people who contributed to this, either in the SOC or in the workshop or through the comment period, thank you so much. This is your project too. So just to get started, you might be wondering, why did we decide to do this? Why do you think we need something like a Biosignature Standards of Evidence activity? And really, this is because we're recognizing that the detection of extraterrestrial life, which may be fairly imminent. We have so many projects going on. This will be neither certain or this will be an unambiguous thing. And this is a high stakes activity. And so we want to have this way of framing the way we talk about our results so that it's clear to folks what our discoveries actually entail. The next thing to think about with this, oops, I'm not able to advance. Oh, there we go. Is that this is an explicit goal. We do have many activities going on around this. And so we do need to have a way to convey the complexity of our multi-step continuum process that leads us to make the interpretations we do with the measurements we make. Now, there was a similar activity like this that was done by a number of folks at headquarters that resulted in a paper that came out in Nature Communications last year, just after our workshop concluded. And that was led by Jim Green. And you'll see some similar themes in what people talked about in the workshop, even though there wasn't necessarily overlap with that group explicitly. The other thing that I would want to point out as why we decide to do this is that, yes, these activities that we're doing and exploring are leading to more and more instances where we have a need to be able to convey our certainty about any discovery. So we have increasing claims of possible life detection. So it's really important for us as a community to come together to talk about how we're going to convey this information in a way that respects the interdisciplinarity of our pursuit. So yeah, the workshop goals were really about how do we make a generalized progressive framework for robust biosignature assessment. And given that we have people who work on in-situ techniques, paleobiologists, and exoplanet people, and people who do just remote sensing, that's why we really needed to have this generalized approach. So we want something that's applicable to a range of detection methods, something that's efficient for the community to implement, not anything that's going to be, too many steps. Try to think about lumping and splitting when we talk about how we describe our activities, and frame the search for life as a continuum of objectives. As scientists, we all know this, that every piece of evidence adds on the next, but it's important for us to be able to convey that accurately. And also talk about what isn't known and what the next measurements we need to do to bring more certainty to our conclusions would be. So in July last year, we held a flipped format, fully virtual workshop. We provided everything that the participants would need ahead of time. That included a whole lot of reading, thousands of pages. Our participants were absolutely heroic, and hours of videos that our SOC members and others provided. The workshop was 3 5-hour days with a dedicated writing day in the middle of all of that. And we got our report writing kickstarted during that. And we also wanted to recognize that all of the authors participated in weeks of focus groups after that. We had 125 participants with 215 asynchronous participants chiming in. And we had a pretty good mix with a little over half being solar system or planetary scientists, 18% paleobiologists, again, recognizing that heritage we have in astrobiology, and a quarter of our folks were remote sensing specialists or exoplanet people. We had international input with the workshop spanning 3 time zones with an APAC group and a Europe group as well. And they contributed and had their own breakout sections and workshop discussion groups. And we brought all of the comments together in the large workshop report that many of you have probably read. So what was this all about? What was the outcome of this? The outcome of all of this was the Biosignature Assessment Framework. It's a 5-step thing that we can look at to think about how certain we are and what evidence we have for biosignature life detection discovery. The first thing, and I'll go through these in detail, the first thing is, have you detected an authentic signal? Second, have you adequately identified that signal? 3, are there abiotic sources for your detection? 4, is it likely that life would produce this expression in this environment? 5, are there independent lines of evidence to support a biological or non-biological explanation? And we recognize that all of these questions will need to have standards of confidence and certainty, adjusting them. And that's work that we plan to do in the future. But the workshop goal is really making this generalized framework and figuring out how we can use it, how to actually apply it. And a lot of what we did in the workshop was challenge the framework with a range of examples. And that's exactly what Erica and Eddie are going to do for you in a minute. So we would like to think that the 5 steps can actually be combined into a model of level 1 and level 2 activities, with level 1 being detection and level 3 through 5 being context and assessment, or level 2, which is questions 3 through 5 being context and assessment. And so that the important thing that I would say about this is that it's not a linear path, that you can continue working within either of these and iterate at different levels simultaneously. You don't need to do one to go on to the next to provide more and more background information for your discovery. I mean, these can simultaneously address questions within the level, and a high level confidence in any detection can then be moved on to level 2 to think about context and assessment. And level 1 activities we like to think of as being, kind of a discipline specialist, whereas level 2 very much is an interdisciplinary activity that needs all of us to think about. So as we move down this framework, we're getting into more and more required expertise. And we wanted to keep this generalized, yes, so the remote sensing and in-situ measurements would be on the same scale, even if they have different confidences. So if you have read the Jim Green paper, you'll know that this first thing, question 1, an authentic signal, isn't necessarily something that's in there. And this is really an example that we like to think about, especially for remote sensing, because there are sources of noise and things like that that would affect your ability to say, is this actually a signal? So that's why we added that in there. Have you adequately identified your signal is really a way of saying, have you considered potential contamination? This is the real contamination question here. And for whatever your technique is, are you considering how you're making your identification with respect to standards and other observations? Question 3, are there abiotic sources for your detection? And this is really a way of getting everyone to start really thinking hard about environmental context. This is probably the most challenging part, I would think, because there's relatively little we know about abiotic processes living on this planet that's just saturated with life. So this really brings in all the expertise of people who just study planetary systems to help us understand these things. If I had a nickel for every time we heard context is everything over the course of the workshop, I could, probably retire right now. That's a theme that kept on coming up over and over again. Question 4, is it likely that life would produce this expression in this environment? This is really requiring us to query this environment and think about what our analogs are that we would have this interpretation and really understand, especially like, for example, it's a paleo example like Eric is going to give us, how much do we know about that environment? If we think we find something that would have been swimming and we know there wasn't ever water there, we should probably rethink our interpretation and maybe go back to question 2 and re-examine things like contamination. So this is another example of really digging into your context and thinking about the preservation of your biosignatures as well. Do you expect them to be in this quantity in this environment over this period of time? Question 5 is about independent lines of support for any explanation that you have for a data set. This is a way of thinking about false positives and what next steps there might be and how you can lump together data sets to support the conclusions that you're making. This is really a recognition that there's no single detection that's going to have the appropriate level of confidence for us to make really robust interpretations, that we really do need multiple lines of evidence. And I think this is actually fairly intuitive for most of us working in the field. And that confidence level can be increased, not just with a single new thing that you observe, but a suite of observations. And this is another way of really getting us to think about how we work together as a community. We had discussions in the workshop of applications of the framework. We want to be able to think about how we place discoveries on a spectrum of confidence, how we convey that uncertainty, how we communicate that uncertainty and that confidence, and really think about how our missions are designed and how the standards of evidence can help us understand assessing the measurements that we would make during those activities and how they really advance our life detection goals, with the understanding that we need so much more context and physical environment information. We're thinking about a couple of different future directions. Like I said, we're really thinking about a confidence scale activity. We're thinking more about mission concept development, ways that we can integrate standards of evidence with things like the life detection knowledge base. A future activity really would be to bring in other stakeholders in these activities, like communication specialists that help us talk to the public about our discoveries that we make. And so those are things coming up. If you're really interested in those topics, those things will need to be discussed within the community over the next few years. So I wanted to just end with a great quote from a paper that came out last year in a completely different field, cancer biology, but is really relevant to our pursuit here. No single effect, experiment, or paper provides definitive evidence about its claims. Innovation identifies possibilities, verification interrogates credibility, and progress depends on both. So I'm going to hand it over to Erica and Eddie, who are going to go through some examples of how we use that 5-step framework in the context of data that we analyze.
Excellent. Thank you, Heather. And I always love thinking about it, the quote that you brought up, the how excited should we be about this discovery? Because that's what it all boils down to. We all want this. And it's one of the most exciting things that we can think of. But like, we want to try to get the scientists throughout the world, all the expertise that we don't have, and get together and figure out, should we be excited? How excited should we be? So thank you. As she said, our next speaker is going to be Erica Barlow, and she's going to be presenting a worked example, because we find that taking these concepts through from the workshop and showing it in practice on how we actually interrogate discoveries and different scientific information we have out there is one of the best ways to demonstrate this in practice. And so a brief intro is Erica is a NASA postdoctoral program fellow in Professor Chris House's group at Penn State. She obtained both her Bachelor in Environmental Science degree and her PhD in geology at the University of New South Wales, Sydney, where her research focused on morphological, textural, and isotopic biosignatures in the early Earth rock record. Her current research explores the idea of past agnostic biosignatures to contribute to the development of tools to be able to recognize and describe fossilized life as we don't know it elsewhere. So if you're ready, Erica, feel free to share your screen.
Thanks for that introduction. As soon as I shared my screen, I lost the controls to unmute, but can I just confirm you can see the slides all okay?
It's great.
Yeah, it's good.
Great. So yeah, thanks for inviting me to present on this worked example. It uses stromatolites from the early Earth rock record to show how the framework steps could be applied to assess an in situ detection of a laminated stromatolite-like structure. So firstly, just so we're all on the same page, stromatolites are laminated sedimentary structures that form at the sediment-water interface in a range of environments as a result of the influence of microbial growth and metabolic activity. And stromatolites are one of many types of microbialite, which is a broader overarching term that describes deposits that have been formed by a combination of biological and sedimentological processes. So for this worked example, where you've detected a laminated structure that you suspect may be biological, question 1, have you detected an authentic signal? And question 2, have you adequately identified the signal? Were considered pretty similar. So basically, are you sure you're looking at what you think you're looking at? Clearly, there's some kind of structure there in question that, that's been identified, but is it actually embedded in the rock, or is it only preserved on the outside? Can you see it penetrate in 3 dimensions? What's its composition? Are there veins present? What are the relationships between potential veins and layers? And all of these questions can be addressed through observations in the field and thin section petrography to help kind of give you confidence that you've actually detected something real. So in doing this, in these field observations, it's important to look at multiple scales because stromatolites occur across basically every size that you can think of. So in the top left here, we've got really broad, multiple meter scale domal stromatolites. And in the top right, centimeters sized. Bottom left, is millimeter size, really tiny microdigitate stromatolites. You can see the tip of a finger there for scale. And in the bottom right, micrometer size stromatolite in a thin section slide under the microscope. So I guess observations of the morphology and the texture and the composition need to kind of be looked at these multiple scales. As one example, you can investigate the relationship between your laminated stromatolite-like structure and the surrounding layers. So taking an example from these stromatolites here from Shark Bay, you can see that the sediment indicated by the orange lines abuts up to the edge of the stromatolite mounds. So when you look at this kind of relationship in a fossilized example as a cross-section, the stromatolites here are outlined in blue, and again, the sediment layers are in orange. And you can really see that these two, although they're both laminations or layers, they're really actually distinct features. So many stromatolites exhibit, I guess, what we call vertical relief above the surrounding sediment, and this can be reflected in the fossil record. So it's something to look out for. Another feature that you could consider is whether there's convex laminae and also potentially the thickening of this laminae over crests, because this could be indicative of phototrophic behavior. Another feature could be overhangs that maintain their shape, because this could be explained by the cohesive properties of microbial sediment. Another feature to look out for in the field is whether the structures are both vertically and horizontally extensive throughout your stratigraphy, because stromatolites commonly grow in clusters or bioherms with many individual specimens preserved in close proximity. And another feature is if your laminated structure kind of develops and changes, this could support biogenicity. As an example here from the Tambiana Formation, you have these really beautiful wide columnar stromatolites that then progress upwards and change to like these like smaller laterally linked kind of finger columnar stromatolites. So if there are multiple types of features that look morphological, that look biological in close proximity, that's going to help support your biological interpretation. So question 3, are there abiotic sources for your detection? So an example of this might be deformation or folding of sedimentary rocks, which can broadly mimic the morphology of stromatolites. So in order to assess the likelihood of this, it's important to look at the deformation and metamorphic history of the rock unit. Of course, this question is building on all of that field work and observations that you've already kind of collected in steps 1 and 2. Another example of something that like an abiotic kind of mimic could be crystal fans. As you can see, they broadly kind of mimic the shape of stromatolites, but you can generally differentiate these through observations in the field and looking at the mineralogy in outcrop and in thin section. You can see that the horizontal layers either side of these fans, you can actually trace these through the fans and the other side. So it's the sediment that's been displaced by these crystal fans. And another example of a potential abiotic mimic is these mud cracks, at least in plan view, could look like lots of stromatolites packed in together. But if you investigate in 3 dimensions, you'll see that the mud cracks don't actually penetrate very far. So basically, you can differentiate between abiotic mimics and stromatolites or laminated, potentially biologically laminated features in the field, and then also taking samples and looking in petrography and the composition under the microscope. So step 4, is it likely that life would produce this expression in this environment? Here, it's important to investigate the regional and local geology to uncover the environment of deposition. So basically, is the environment and the laminated structure itself sedimentary? And could life have lived here? And could it actually have been preserved in this environment? So here's a diagram that shows multiple stromatolite morphologies along the bottom. And then these are tied into the interpreted water depth. So on the left is shallower water deepening towards the right. And you can see that the morphology changes according to water depth and also the energy of the portion in the system. So when you're looking at your laminated structure, you can assess whether the shape of it varies and whether this change is consistent with the physical and chemical characteristics of the paleo environment, including, for example, the interpreted water depth. And another thing to consider in this point, I guess, is the preservational angle. So is it plausible that life could have been preserved in this environment? Is there an apparent mechanism for preservation, such as early diagenetic silicification or carbonate precipitation? What's the means for actually preserving the structure that you're seeing? And so step 5, are there independent lines of evidence to support a biological or non-biological explanation? So I guess steps 1 through 4 outline some of the essential lines of evidence in establishing whether a laminated structure is biological in origin, but there are additional lines that could be used to further strengthen a biogenicity claim. So these could be things like the presence of syngenetic, biogenic organic material detected through Raman or other laboratory analyses, stable carbon isotope data, especially if this is in-situ analyses tied into the specific laminae that you're looking at as opposed to a bulk analyses. It could be other isotopic evidence from minerals, for example, sulfur isotopes from pyrite, and these could indicate a biological influence on the mineral formation. And there could be other morphological signs of life, such as microfossils, other microbialites, or microbially induced sedimentary structures, just to name a few. So to bring this all together, here's a summary diagram of the framework steps and some of the necessary actions at each step. I won't go into the detail because I've briefly kind of covered this, and also there's more info about it in the workshop report. So feel free to go and check it out in more detail. But I guess to summarize, if you're just looking at an individual line of evidence, there might be a fairly convincing abiotic explanation for it, but it's unlikely that multiple lines of evidence supporting the biogenic origin will exist in the same sample in the absence of life. So as Heather drove home earlier, multiple independent and complementary analyses are key to assessing the validity of a detection and to improving the confidence level of the biological explanation or non-biological explanation, either way. So thank you very much. I'd like to acknowledge the workshop organisers and also the Early Earth Writing Group that helped put together this section of the report.
Thank you, Erica. It's always exciting to think about actually being on site on like Mars or some other system and being able to see these structures directly. That's the dream. But now to switch to a completely different scale, we're going to get a worked example from Eddie. So for some background, Edward Schwieterman is an assistant professor of astrobiology in the Department of Earth and Planetary Sciences at the University of California at Riverside. He was previously a postdoctoral fellow at Georgia Tech and a NASA postdoctoral program fellow at UCR. He received his PhD in 2016 at the University of Washington with dual titles in astronomy and astrobiology. He completed his undergraduate degrees in astronomy and physics at the Florida Institute of Technology. And he has spent 2 lovely summers researching and observing comets as an undergraduate at Lowell Observatory in Flagstaff, Arizona. Take it away, Eddie.
Thank you very much, Bradley. So as I start, I'd like to acknowledge the contributions from the writing group that looked at oxygen as an exoplanet biosignature. In particular, Sonny Harmon and Joshua Sanson-Totten and Vicki Meadows and I contributed to this report in addition to many others. So I'd like to start with Earth spectrum, reflected light spectrum today, and its features. So what do we mean by reflected light? Reflected light is the light that comes from the host star, is reflected or scattered by the planet. And then we can split that light apart with a prism, or more accurately for a space-based telescope, a diffraction grating, and look at its spectral features. So that is the influence the atmosphere or the surface has on the light that's been processed by the atmosphere and reflected or scattered to the distant observer. We chose oxygen as our example biosignature because on Earth, it is the ultimate expression of the oxygenic photosynthetic metabolism on Earth, which is by orders of magnitude, the most productive metabolism on our planet. It has drastically changed our atmospheric composition and it produces observable features. We look at the reflected light observing mode because that is the best observing mode to detect oxygen. And it is also the consistent with the architecture for the telescope that's been recommended recently by the Astronomy and Astrophysics 2020 Decadal. So what I'm showing in the slide is an Earth average spectrum from the ultraviolet to the near infrared. On the x-axis is the wavelength in microns. So you multiply it by 1,000 to get nanometers. And on the y-axis is the geometric albedo. So that is the brightness of the planet divided by the host star. And what you can see here are features from oxygen, water, ozone, and a little bit from carbon dioxide and methane. The large ozone feature here, that's the Hartley-Huggins band. We're happy to have that on our planet because it gives us our protection from the harmful effects of ultraviolet radiation. And by definition, then also imparts an immense observable signature to a distant observer. So the idea is starting from here, how are we going to verify that this oxygen is indeed biogenic? But first, it's important to know that this is not what we'll actually get if we're observing a distant planet. In fact, what we will actually get is a much more noisy spectrum. So on the left is a simulated coronagraph observation of the solar system from a distance of 10 parsecs. What's important is that either a coronagraph or star shade instrument needs to be used to null the light from the star. The planet, in this case Earth, is only about 1/10 billionth the brightness of the star in the sky. So this sort of nulling is essential. On the right is a realization of the noise of an Earth around a nearby star, Beta CVN. This is from the HabEx report. And what you can see is the input spectrum in sort of this purple color, and then the simulated data points in yellow and gray. And so this is more accurately portraying what we're actually going to receive from an exoplanet. And this green dash right here is indicating the strongest oxygen band in Earth's reflected light spectrum. That is the 0.76 micron oxygen A band. Okay, so how do we characterize this oxygen in the context of the 5-step framework that's been presented by Heather and Erica and applied to stromatolites, and now we're applying it to oxygen on a distant planet. So we really need to know that this signal is in fact detected. So one sort of boundary we need to get across is the signal to noise ratio adequate? Often for remote observations, that threshold is about a signal to noise ratio of 5. We need to exclude instrumental systematics. So for example, the noise that's a result of the intrinsic characteristics of the instrument, the noise that's introduced by the star, so stellar variability is key here. Remember that the star is in fact outshining the planet by a factor of 10 billion. And indeed, the light that we're receiving from the planet originally came from the star. So characterizing stellar noise is incredibly important. We can help confirm this oxygen signature by detecting oxygen at multiple wavelengths or multiple epochs, so at multiple different times. And that would strengthen the detection. So there's another oxygen band in this wavelength range, a weaker one at 1.27 microns. There's also one at 0.69 microns and at 0.63 microns. Those are fainter and less easily detectable bands, but would be consistent with the primary oxygen detection at 0.76 microns. So step 2 is the feature sufficiently identified as O2. So one way we could potentially, sufficiently identify this signature as O2 is again, detecting oxygen at multiple wavelengths and multiple epochs. The other way is looking for a well-known photochemical byproduct of that oxygen, which is ozone. So ozone is anticipated to exist just by nature of the spontaneous photochemistry that will happen in the upper atmosphere of the planet. And ozone indeed creates large features in the ultraviolet spectrum, this Hartley-Huggins band. And so detecting what we think is oxygen along with what we think is ozone would further strengthen the identification and of that oxygen band initially. And so there are two here. There's also this Chapuis band from about 500 to 700 nanometers with a maximum about 575 to 600 nanometers. So that would help confirm that we've indeed detected the oxygen. Another thing that will help is applying atmospheric retrieval models. So an atmospheric retrieval model is an inverse model that is informed by a large population of forward models that exist as a library. And from this retrieval model, a statistical estimate can be made for the abundance of gases that are detected in reflected light, but also the major planetary parameters, such as the radius of the planet and the surface gravity of the planet. That'll be important later when we think about context. So if we're sufficiently convinced that we've actually detected oxygen, we also need to determine whether that oxygen is in fact produced by biology, or if it's consistent with production by biology, or if there are abiotic processes that are likely to have detected it. And so there's a large literature that exists, and I've only cited a small amount, that have posited different mechanisms for how oxygen could be produced abiotically on a terrestrial planet. Some of these abiotic production mechanisms are most efficient around certain types of stars. So this is where context is important, characterizing the host star. So for example, M dwarfs have extended pre-main sequence lifetimes. So any planet that is within the habitable zone today has gone through substantial water loss if it's always been in that portion of the habitable zone. And that is the photolysis of water and the escape of hydrogen and the retention of oxygen potentially. And that would be a much more efficient process than has ever existed on Earth in Earth history for a longer period of time. And so there may be large amounts, or at least it's been hypothesized that there may be large amounts of abiotic oxygen on M dwarf planets. So one way to get around that potentially could be to search for evidence that there is too much oxygen. And so that's this middle panel right here. If there is a substantial amount of oxygen, there are certain spectral signatures called oxygen-oxygen collisionally induced absorption. Those are dependent on the density of oxygen squared rather than simply the density of oxygen, and would produce observable bands. And so the detection of those may indicate, in fact, that this is there is an abiotic source of oxygen. Another source of abiotic oxygen is the photolysis of CO2 and the partitioning of that CO2 into CO and oxygen. And this could potentially happen if there are a lack of catalysts that help recombine that CO and O back into CO2. And so one signature you might look for to see if this process is happening is the simultaneous presence of CO and CO2, and maybe the lack of catalysts that would help recombine those two species, including methane, which is a biological product. And in certain cases, if you lack water, that process of partitioning CO2 into oxygen and CO is even more efficient. And so an absence of water may also help indicate that this process is happening. In contrast, if we were to see water, methane, Rayleigh slope consistent with the scattering from Earth's atmosphere, so we're seeing through to the surface, in addition to oxygen and ozone, that may be more indicative that this is not an abiotic source of oxygen. And in fact, it's a photosynthetic source of oxygen as exists on Earth today. So this is really where context is key. Is it likely that life would produce the oxygen? So is the oxygen congruent with the planet? And so in order to do this, we might look for several things. We may look for water. Water vapor would indicate a presence of catalysts that help recombine CO and O back into O2, so that would help. It may also be suggestive of an ocean surface, although it wouldn't confirm it. And of course, water and carbon dioxide are both needed substrates for oxygenic photosynthesis. So the detection of both of them together would be congruent with the presence of oxygenic photosynthesis. In addition, we might look for the evidence of oceans directly. Ocean detection could be supported by multi-phase mapping, where we're looking at the planet as a function of time. And due to that time-dependent photometry, we can therefore extract a rough albedo map, longitudinal albedo map of the planet. At some rotational phases, the planet will be darker and some it will be brighter. And that rotational phase can be connected to the orbital phase. In other words, we would expect to see ocean glint, the detection of specular reflection from the ocean, at the crescent phases. And so if we were to look at the planet near to quadrature and see that there's a darker surface and a brighter surface, that darker surface could be ocean and the brighter surface could be land, we could confirm that by then looking at crescent phases. And is that darker surface now the brightest surface? That would be indicative of specular reflection and the direct detection of oceans. So this has been done in hypothetically with the Earth as an example. So the epoxy spacecraft and other Earth observing spacecraft have looked at the Earth as a function of phase and rotation and applied photometric mapping techniques to extract the longitudinal albedo maps of the planet and create suggestions of the ocean and land presence. In addition, we might look for stochasticity that's indicative of cloud cover. And the cloud cover would be consistent with a hydrological cycle. And again, supportive of the evidence that there is land and ocean. And that is congruent with the presence of life. And finally, we're going to look for independent evidence for biogenicity. And that includes additional biosignatures. So oxygen and ozone may be the most detectable signatures. But once those signatures are found, and we've ruled out some of the more common or more easily detected abiotic processes, we might look more deeply. We might prioritize this target to search for less detectable, but more confirmatory signatures. Those include additional biosignature gases like methane or nitrous oxide, and surface features such as the vegetation red edge. The vegetation red edge, of course, is this contrast between the reflectivity of vegetated surfaces in infrared light versus their darker absorptive nature in the visible light. So this VRE signature would also be correlated with land and ocean surfaces and could potentially be extracted with time-dependent rotational photometry. Seasonality is another potential biosignature. So that is that the seasonal changes in gases may also be indicative of a global biosphere. These signatures may be much harder to detect than oxygen itself. But again, the difficulty is compensated for by the relative lack of false positives. So this is vegetation red edge. I mentioned different gases are just some of the possible biosignatures on terrestrial planets. There isn't enough time to talk about all of them, but there are a variety of proposals in the literature. And again, I'll emphasize most of these are harder to detect than the primary oxygen. But once we find that oxygen and we've ruled out the false positives, now the calculus has shifted, and we're really going to want to spend more time on this particular planet because it has the greatest potential for a positive confirmatory biosignature. So the framework in summary is here. We have both the level 1 and level 2. Level 1 is on the right, on the left, and level 2 is on the right. Context is important. Identifying the signature is the first step. Confirming it's the gas that we think it is the second step. And now we go through a kind of a long and iterative process of ruling out the false positives, demonstrating congruence with the biosignature in the planetary environment, and potentially getting confirmatory biosignatures that confirm the biogenic nature of the detected oxygen. I'll note that similar types of frameworks have been endorsed by the LUVOIR and HabEx concept reports. These are concept missions that were proposed to the National Academy of Sciences prior to the decadal report that would use reflected light to try to characterize terrestrial planets. And they have similar ways of ordering this and prioritizing these observations from the context up through detecting habitability, through detecting our primary biosignature, and then our confirmatory biosignatures. And then I'll end just with an importance of observing mode. We discussed reflected light in this example, but there are other observing modes for terrestrial planets, including thermal emitted light, so thermal infrared, which would give you a different suite of potential features to characterize, and transmitted light, that is light that's filtered through the atmosphere as the planet transits. And those signatures, again, would be different in magnitude and quality than those that we discussed here with the reflected light example. So that is my presentation, and I'd like to thank again the contributors to this worked example.
Thank you very much, Eddie. And I think we'll just jump straight into questions to make sure that there's ample time for discussion. So the viewers here, you can feel free to unmute yourself and ask the questions directly or through the chat. So I'll kick it off with a question from the chat from Alex Pavlov. So if a stromatolite structure satisfies conditions 1 through 4, but lacks any organic signature or isotopic delta C13 feature for number 5, then do we mark such a discovery as inconclusive?
That's a really good question. And I'm going to give a somewhat annoying answer. It really depends on the actual, example that you're looking at. For instance, if you took pretty much any of the stromatolite images I showed, I would be very, very confident in the absence of organic material and in the absence of carbon-13 isotope signals that those were biogenic. So it really depends on the example you're talking about, the context of the system. But I guess one thing to say is that there are fossils that, we don't, through, the fact of being metamorphosed, very old, don't necessarily have that organic material left there preserved. And that doesn't mean that they're not, or that they weren't fossils, or that they're not life. So I guess the absence of that doesn't preclude it being life.
Yeah, I think that's a great way to generalize it, Erica, that all of these added lines of evidence, it's an additive activity. It's not necessarily going to take away from the fact that this is an interesting feature, that we don't have great physical ways of describing how it would form. And yeah, lots of fossils don't have organic material left in them or any way to interrogate something like isotopes, but that's not the last tool you would ever use to try and get another line of evidence. You could use, co-precipitating minerals, things like that. There are lots of ways to try and get that additive evidence to raise the level to which you are confident. But it doesn't take away necessarily. Yeah, great answer.
Yeah, that's a good point.
I guess you, the viewers, can tell us if we think it's a great answer. You can tell us.
Excellent. We just had a question come up from Mark Elowitz. Are there any plans to search for so-called technosignatures? Technosignature gases, artificially produced gases in exoplanet atmospheres, such as CFCs, HFCs, SF6, NF3. Such a search should also be carried out on planets that may not be Earth-like, given that advanced civilizations may simply use some planets as servicing worlds.
Yeah, I can answer that. So the answer is yes, this is something that's being thought of. There is a workshop, there's a paper coming out of the TechnoClimes conference that was held in fall of 2020. That's in revision right now, that discusses some of this as a review. But I'll note that the technosignature gases that were mentioned are poorly characterizable in reflected light. And so we would not really be able to see them in the same kind of wavelength range and with the same observing mode in which we look at, where we're looking for oxygen, for example. So we need to look in the infrared.
Thank you, Eddie. I had a specific question or a comment that I'd like to hear as well. So oftentimes from the outside, these seem like two very disparate communities, the in-situ and the remote sensing. So I'd love to hear what you've discovered through the process of working together on this project and how they contribute to each other for life detection. Why make this workshop in the first place?
Yeah, I think that was such a big challenge of the workshop. And Eddie and Erica, I don't know how much time you spent with each other in any breakout groups. So it would be interesting to hear how you were able to align your pursuits. But I think this is a great example of how at its core, much of the work we do in science can be generalized regardless of the data type that you are collecting or the environment in which you're collecting it, that it really comes down to how you're approaching your questions and how you're crafting your interpretations. And that's what, we think this 5-step assessment framework can help all communities actually do. I think one really interesting thing, and I kind of brought this out a little bit, is the fact that we really did try to address the fact that much of our information is being mediated through a technology, and that technology has sources of error or noise or uncertainty inherent just in its construction and design. And that becomes that's a very big example from something like remote sensing, but we really need to think about it hard when we think about in situ applications. And I have to say, I was really impressed with how the early Earth group that Erica, let us know about, addressed that themselves to try to think about like, what is the noise when I'm observing something, regardless of if it's, contamination from metamorphic activities or something like that, but are there ways that we're taking apart a rock that are affecting our interpretation, things like that. And that's summarized in the report as well.
Thank you, Heather. Are there any questions from the gallery here? Any further questions?
I would just add on the last question. I think both of us came in with a perspective from the In-Situ and the remote community that the other community had the harder job. And then I think through discussion, we realized that. And I think it was useful from the perspective of really understanding the challenges that exist between the communities and putting them in a new light.
It comes up from time to time, so I just thought I would reiterate, since I didn't say it explicitly in the intro too, when we talk about generalization, this is very much a framework that allows us to be agnostic about our discoveries as well. There's nothing in those 5 steps that say that you necessarily have to be looking at an analog of life from Earth. We say things like standards, but that's really about understanding the data that's being generated by any of your instruments or any of your measurements that you're making. That's not saying you have to assume something. I wanted to point out how this fits in too, is that Sanjoy said something really astute in the chat with respect to Erica's example, that, making a carbon isotope measurement does not actually tell you anything about the biogenicity of something, because again, like question 3 in the assessment framework, you have to have that context information to tell you if there's been significant fractionation that we would associate with a biological process. So you couldn't just look at some rando fossil, look at the carbon isotopes that might be preserved in that and say something about its biogenicity. You'd have to first understand the system, the source pool that carbon came from for that potential organism.
Yeah, and that's a really good point. I'll add as well that I guess as a related thing that your carbon isotope value is hooked into, I guess, what we know about life here on Earth. So even if we found something on Mars or somewhere else and we start analyzing it through carbon isotopes, that isn't necessarily, I guess, calibrated with our system here or life here. So you really have to explore, that whole reevaluate that whole system and understand the context of the planet that you're looking at.
This is Dave DeMarais. I just want to extend that comment to stromatolites themselves, and that is virtually all those structures represent a very tight interplay between the sedimentologic processes and the biological ones. And so it really underscores that you need to characterize the sedimentological processes. It's right in canyon with the biological measurements. Again, this importance of context.
Thank you.
Absolutely.
And
thank you for that comment, Dave.
I'll highlight 2 comments as well, and then go on to a question. So Nikki Parentos pointing out, likes to reiterate, when you're reporting these results, this doesn't prevent you from reporting on any findings. It just helps to guide you on communicating where you are and your level of confidence on the scale itself. And then Mark Elowitz says the remote sensing community is vital since many of their members have significant experience in the use of hyperspectral imaging spectroscopy data analysis in support of Earth observations, and the Earth is simply another Earth-like planet. Many members of the terrestrial remote sensing community have experience analyzing and simulating spectral data over the wavelength range 7.5 to 14 microns, where many fundamental vibrational modes are present from various gas molecules of interest to the astrobiology community. And then the question from Daniel Wan, Are there any particular drives for the investigation of biosignatures that arise from alternative biochemistry?
I feel like that's
like
a question for you, Heather. Yeah.
I was about to say that's a question that probably either me or Eddie could take on. And I think what you're asking there is if we can, model or imagine some metabolism, and then, try to think about what those products would be, and then design a way to try and look for them. And I think this is a place where thinking about what is likely in an abiotic from abiotic processes in any environment is really important. Looking for something that is unlikely in those environments. So really, again, back to that context is everything could be a way of addressing that same thing. I don't know, Eddie, is this something that you guys think about with remote sensing as well? Do you have a more substantive example than I just gave?
Well, in order to detect something remotely, it has to build up to concentrations, adequate concentrations in the atmosphere. So it needs to be robust to photochemical reactions or photolysis. It needs to produce features that are detectable given your observing mode and instrumentation. So whether you think about reflected light or transmitted light or emitted light. The sort of answer to that is there are a lot of unknown, you have to stack vertically all of these factors. And so when you start with unknown biochemistry, it becomes difficult to then build out that detectability analysis. On the other hand, it's a good point that we may find things we do not expect. And I think ultimately we may end up discovering things on planets that then force us to go back to the drawing board and think about, how do you explain the signature? So the signature will be something unknown, but it's very difficult to start with, how do we find life as we don't know it, and no data, and then create, a self-consistent story. Some people have looked into that, and it is, a worthwhile thing to do. I think ultimately, right, we're going to have to wait for the data in order to really make advances in that area.
Yeah, and I think a great way to contextualize this as well is that question 4, would life be making this in this environment, given this source of materials, given this energetic, system that you're looking at, would this be a likely outcome? Is another way to try and tie in that otherwise you could look at anything and say, oh, that might be life. And this is really getting to the point, is life your first or second or last hypothesis when you're looking at anything in nature, especially, in this context.
Can I add to that? I think one of the huge advances that the exoplanet people have made is looking at discovering all the ways oxygen could be produced abiotically, and then reconciling that with the environment of the planet. And at Mars, it's the same thing. We were going there trying to characterize the environment. And the next step is, okay, given what we know, are there abiotic means of creating features that might be tempting as biosignatures? So environmental characterization, processes associated with those environments is a necessary prelude to really trying to get serious about recognizing biosignatures, particularly ones that are not, totally mirrored on Earth.
Okay, excellent. Thank you for that comment too. I agree. It's time to wrap up. We've crossed the top of the hour. So thank you again, Heather, Erica, and Eddie for this. And as a heads up for everybody out there, next month for this webinar, we are also going to be talking standards of evidence as a lead up to a town hall discussion that we are going to have at AbSciCon. So you can definitely get your standards of evidence fill for the next 2 months. It's a big topic of discussion that's made a lot of ripples in the community. So we want to make sure we have time to address it and to get feedback from all of you. So thank you all for attending, and look forward to seeing you next month.